talking about mitosis. This is part 2. We're focusing on mitosis as it relates to cancer. And so let's get started. Mitosis and cancer are connected to each other because cancer is growing out of control, or cells that are growing out of control. And cancer cells can actually produce something called malignant tumors. So cancer cells are cells that are dividing excessively through mitosis to form masses. And those masses are what we call tumors. If a tumor then has a cell break off and invade to a neighboring tissue to then make those cells divide out of control, we call this a malignant tumor. When we talk about how normal cells exhibit density dependence and anchorage, where they need to actually be in contact with something, there is a limit to the cells that produce growth factors. Those are all things that have normal cells. Cancer cells do not. Cancer cells result from mutations in genes that control the cell division and that allow them to divide uncontrollably. And so that allows there's mutations in the genes that then produce proteins that affect the cell cycle that allow the cell to go through mitosis over and over and over again, ignoring density dependence, anchorage dependence, or a limit of growth factors in the area. Now, proto-oncogenes are genes in the body that normally help make proteins that control cell division. They could become oncogenes, which are cancer-causing genes, in 3 ways. 1, they can make a hyperactive form of growth-stimulating hormone. We can make too much growth-stimulating hormone either by moving the gene to a different location so it becomes expressed over and over again, or we can overstimulate the cell division. So again, a proto-oncogene is a gene that has the potential to become an onca or cancer-causing gene that then is directly involved in cell regulation or the cell reproduction regulation. All right, so tumor suppressor genes are mutations that when they're inactive, they suppress genes, and they have a similar effect to oncogenes. So if cell division is controlled, then the protein is great. If it's defective, then the cell cannot control cell division. So for example, the p53 protein will stop a cell from dividing if there is damage to the DNA or there's a problem in how prepared a cell is. If p53 is mutated, then the cell is likely to undergo uncontrolled cell reproduction, which is cancer. Now, p53 mutation is found in approximately 50 to 60% of all cancers. And another type of protein that is found in cancers is called the RAS protein. So both p53 and RAS proteins are, if they're defective, they can have a direct link to cancer. So oncogene proteins and their kind of faulty tumor suppressor proteins can directly interfere with the normal signal transduction pathway where we have DNA, RNA protein, and that's being regulated by several different factors. Now, the RAS protein is a relay protein that stimulates cell division. And if the RAS protein is hyperactive, then it causes too much cell division, even without any growth factors, triggering the phosphorylation cascade or triggering the transduction signal. So here we can see in the diagram, we have a growth factor protein. It binds to a receptor protein on the membrane, triggering our RAS protein inside. And that causes all of our cell division proteins to become active and the cell will divide. Well, even without the growth factor, if the RAS gene has a defect and it's constantly going, we'll constantly be triggering cell reproduction or mitosis. Now, p53 does the opposite of RAS and it actually inhibits cell division. So if p53 is mutated, we can't stop cell division, even if there's no growth factors or signal proteins are saying, stop, there's a problem. If p53 can't do its job, we can't stop cell reproduction from occurring. Now, there's multiple genetic changes that actually underlie cancer. And so cancer doesn't just result from one genetic change, it's often multiple mutations, as we can see in this quick little diagram. Here we can see colon cancer actually develops from multiple steps. So eventually there's like an oncogene becomes activated, then maybe a tumor suppressor gene becomes inactivated. So now we're triggering cell division and we're unable to prevent or stop it. And so we get these big tumors or mass that occur because the cells have been dividing out of control. The accumulation of mutations often leads to cancer. So first we can see there's 2 mutations that cause the cell to divide more rapidly. Then maybe there's a mutation that takes the cell and it causes it to break off and go look for a new place. Once it lands in a new tissue, it then triggers those other cells to start dividing out of control. So again, multiple mutations are the trigger for cancer. There is another thing that can happen or cause cancer, and cancer is often caused, again, from multiple mutations. And one of the things that we look at is something called a telomere. The telomere is the region of the DNA at the very end of chromosomes. So it's kind of extra sequence. And it turns out it's actually just a repeat of the TTA-GGG sequence. And every time a cell divides, a person loses on average the last 30 to 200 base pairs at the end of their chromosome because of how our replication mechanism attaches. And so a normal cell can divide 50 to 70 times. And every time it divides, that telomere region gets a little bit shorter. Well, when the telomere is actually gone, then we start losing sections of DNA of the actual DNA material that we need in order for a cell to properly function, which then can trigger those mutations in the genes that help control cell division. One of the things that we want to kind of briefly mention is that Marie Claire King is a female scientist who discussed how mutations can cause breast cancer. And researchers have actually gained a lot of insight into breast cancer by studying families, by looking at the genetic component, by showing that most cancer is actually non-hereditary, but there are some cases in which it is. One of the things that we can do about non-hereditary cancer is we can actually avoid exposing ourselves to carcinogens, and we can reduce our own risk of cancer. What is a carcinogen? It's something that causes cancer mutations. So for example, if we look at this next chart, we'll see that there are carcinogens in things like smoking, or chemical exposure. And it turns out that when we look at the number of cases across the US, the highest rate of cancer is in prostate and breast. And you can see that lung cancer, the #1 risk factor is smoking. There are other risk factors for other types of cancers like viruses, hormones, obesity. And so we can control those factors, and we can't just blame DNA. All right, guys. So what happens if you have cancer? Well, it turns out that radiation and chemotherapy are really effective treatments that we have because they stop cell division from occurring. And again, cancer is a out of control cell reproduction. So again, I want to make sure that we kind of review the functions of mitosis, which is growth, cell replacement, and asexual reproduction. So when a cell operates normally, we're doing growth, we're doing replacement and damage of lost cells, and we're allowing a cell to reproduce itself through asexual or cloning reproduction. And that's it, we're done.